Micro folks, these are lab results for Chapter 17, Metabolic Test Part 2. Part 1 of Chapter 17 was the IMVIC test. Now we're going to do the triple sugar iron slants, and there's a lot of information with them. So let me just show you this cool photograph off of the internet. So you guys, so first of all, triple sugar iron, triple sugar means there's 3 sugars present: glucose, sucrose, and lactose. So we can detect whether one or more of those sugars is fermented, and the way we'll do it is by including phenol red, the pH indicator. So if a sugar is fermented, acids will be produced, and the phenol red will turn yellow, telling us that acid was produced. If the microbe can't use a sugar, it has to tear apart the amino acids, releasing the amino group as ammonia, a weak base. The pH will go up, it'll become alkaline, and the phenol red will turn a deep red color, often called fuchsia. Now, in addition to the acid production from fermentation, we can also look for gas production. So here you guys, we can see in this tube, there's a big old gas bubble right there. And in our tubes, you guys, it's phenomenal, the gas production. So this is our E. coli, 48 hours. You can see so much gas was produced, it's blown the slant apart. And here's our Citrobacter. It also has blown the slant apart from from gas production during sugar fermentation. And the gases, folks, would be molecular hydrogen and or carbon dioxide. I'm guessing both Citrobacter and E. coli are making both molecular hydrogen and carbon dioxide. There's so much gas there. Okay, so we can see sugar fermentation with acid. We can see sugar fermentation with gas. The way we score the TSIs, folks, is we look at the color of the slant and we look at the color of the butt. Okay, and believe it or not, there is some reason to this. So you guys, let me start with this one right here. You guys see how the butt is yellow. So we have a yellow butt, an acid butt, but the slant is alkaline, it's red. So we score them slant first, you guys. So this is an alkaline slant, so we use K. Then we go with slash, and then we give the symbol for the butt. And so alkaline slant, K slash, the butt is acidic, so we have an acidic butt. This pattern, you guys, tells us only glucose was fermented. So if the butt's yellow, that tells us glucose was fermented. If the slant is not yellow, that tells us the microbe could not ferment lactose nor sucrose. So you guys, we go K slash A, and let's see here. This is saying that there's gas produced, and I think it's hard for us to see, you guys, but they must have. Oh, right. So here I'm putting question mark for gas because I can't see if there's a bubble or crack there. That might be a little gas bubble right there. Okay. So a little bit hard to interpret the gas. Okay. You guys, let's go over here to this one. So if we score, this is a different microbe, and you'll see how the slant is acid, the butt is acid, so we go A slash A. Acid slant tells us that lactose and or sucrose was fermented. Acid butt tells us glucose was fermented, and here we can easily see, you guys, that a lot of gas was made. Okay. Over here, folks, let me do this last one here. So, folks, this is another organism. This might be, for example, Pseudomonas. And doggone that Xfinity advertisement. I hope you aren't seeing that. Okay, so you guys, this one over here, alkaline slant, so K slant, alkaline butt, so K butt. So the alkaline slant tells us neither lactose nor sucrose was fermented. The alkaline butt tells us glucose wasn't fermented. So this little guy couldn't ferment any sugars, right? So this could be, for example, Pseudomonas. And if no sugars were fermented, we shouldn't see any molecular hydrogen nor CO2, so we're going to say gas negative. Now, another thing that TSIs can tell us is whether a microbe can make hydrogen sulfide. And you guys might remember from lecture, hydrogen sulfide, it's a cytochrome inhibitor, it's toxic, it could kill us. You know, our strict or obligate aerobes, shuts down aerobic respiration. Hydrogen sulfide also smells horrible to us. It smells like rotten eggs, and maybe there's a survival advantage to that. If we smell rotten eggs, we'll run away from it, right? But to determine whether our microbes have made hydrogen sulfide, and there'd be 2 ways they could make it, folks. There's sulfates in the TSI medium. So if the microbe can carry out anaerobic respiration using sulfates as an alternate terminal electron acceptor, they will produce hydrogen sulfide. as an end product of anaerobic respiration, and or if they can tear apart sulfhydryl containing amino acids such as cysteine, which is present in the medium. Okay, so we don't want you detecting hydrogen sulfide production by taking the caps off the tubes and taking a big whiff to see if you can smell the rotten eggs of hydrogen sulfide. That's toxic, right? So what they've done is they've incorporated iron salts into the medium. That's why it's called TS for triple sugar iron salts. And what's so cool, you guys, about iron salts is that if your microbe makes hydrogen sulfide, it combines with the iron salts to make ferric sulfide, iron sulfide. And iron sulfide, you guys, is a solid black precipitate. So if you see any black anywhere, that's telling you your microbe made hydrogen sulfide. So you guys, if we were to read this tube, then we've got an alkaline slant, so that means no lactose nor glucose was made. We can't tell the color of the butt, right, because there's so much iron sulfide present. That iron sulfide told us that our little guy made hydrogen sulfide, so there's that black. And this is just like, you wouldn't have to remember this, you guys, for the lab exam, but if you have this much hydrogen sulfide produced, this much iron sulfide present, you just memorize that there was glucose fermentation. That's a little detail you don't even have to worry about, folks. Okay, so the TSIs can give us lots of information. So folks, here on our E. coli, we have, it's hard to see because it got blown apart, but we have an acid slant that tells us the E. coli could ferment lactose or sucrose or both. We've got an alkaline butt that tells us that glucose was fermented. We got massive gas production, right? Massive molecular hydrogen or CO2. But because there's no black, you guys, we know that E. coli doesn't make hydrogen sulfide. Now, Citrobacter, a cousin of E. coli, looks really similar. The reason we inoculated Citrobacter is it should make hydrogen sulfide, but we can see after 48 hours, I don't see any black here. So I'm going to take this home and we're going to take a look at it in another couple of days and see if we can find any hydrogen sulfide. Maybe actually I'll leave them here and we'll take a look at them on Tuesday. Okay. All right, folks. So that is that for TSIs. Oh, one more thing, you guys. If you over incubate them, if you leave them in the incubator too long, like 7 days, they'll use up all the sugar, they'll start starving, and then they'll start ripping apart the amino acids. So you can get a false negative sugar fermentation on these guys if you leave them in the incubator too long. Okie dokie.